其实大概只要300几个字，然后呢，有说这样太少了，这样代表说那时候不在天就几个人，然后想说，然后在我大概
And we will also talk about how we're going to grade this information. And then my fault. Sorry. And then we're going to do a short lecture. It's on stem cells, and it's very important. It's had a great impact in the U.S. And other countries in the world. And what we want to do is talk a little bit about what stem cells are and why there would be an impact, why that would make somebody feel so upset about it all that he, and I'm talking about President Bush, would say we're not going to spend any U.S. research money on stem cell research. So those are the 4 things that we have to do, and let's start out with quiz 5. Steve Jobs, Patrick Swayze died of pancreatic cancer. What is unique about pancreatic cancer? It is a case of uncontrolled cell division. That's true, but it's not unique. It's almost 100% curable. That's definitely not true. It's confined to the liver. That's not true. It's the pancreas. Victims almost never live more than 5 years after the discovery of the disease. That was the correct answer. Second question, plants get cancers also. Like human cancers, these plant cancers can be, but aren't always, just undifferentiated masses of cells. Remember we saw examples of that. In some cases, there's uncontrolled cell division, that is to say, uncontrolled by the plant, but not necessarily totally uncontrolled, because wasps and bacteria and others can control it. But the organism that has the gall has got a problem. Are never initiated by viruses, not known, always naturally cure themselves. That we didn't know anything about. Grow because the cells grow, not because the cells divide. That's an interesting question in certain circumstances. Didn't have anything to do with this. Cell division always preserves all the genes in the daughter cells, even the genes that won't be functional in the new cells. The following is evidence that this is true. I felt a little uncomfortable about this question because although I intended to talk about this specific example, I didn't do it but most of you got it right in any event. DNA testing in criminal cases works. And what I'm talking about there is the fact that you can take, in the case of rape cases, for example, you can take evidence, samples of sperm, and compare it to cheek cells and determine whether those 2 DNA samples came from the same organism. Why? Because every gene in every cell is preserved. And so that's why that kind of testing works. Cancer happens, kind of a silly answer. Skin cells can be easily transformed into liver cells. Again, doesn't have much to do with the question. Daughter cells are always bigger than the original dividing cell. Again, so the 3 wrong answers perhaps helped a lot of you get this right. And I've just explained to you the reason why A is the right answer. 4, cancer and the immune system are related in that they are both derived from the same organ. Not true. The immune system's only function is to kill cancer cells. That we know isn't true as well. It's one of the things that it does, but certainly not the only thing. Cancer cells cannot damage immune system cells. That's not true either. We know that from HIV/AIDS, that's not true. And the last answer, our immune system almost always kills new cancer cells. And that was one of the lessons that we learned. We didn't we don't even realize that we have cancer because our immune system jumps on it so quickly. And then the fifth, yeah. No, we're okay. Now, yeah, let's. Okay, now let me just talk a little bit about how we're going to use the data that we have from the quizzes that we took, and also from the papers that you wrote. I got quite a few papers, and I'm pleased about that. I haven't graded them. I just received them a short time ago, but I will grade them over the weekend so that everything will be done by Monday. I'm gone on Tuesday, so we'll have to figure out a way that they can get at that information. But let us say, here, just to explain how this is being handled, let us say that this is how it distributes. You have the potential, first of all, we will take out of the system your lowest quiz. So there will be just 5 quizzes, not 6, that go into making this grade. And that means you have the potential for 20 points, 100 points, 20 points per quiz, and 5 quizzes. And let us just say that it distributes this way, and I then will decide how the grades distribute, and this is one way that they could. Again, I haven't made these decisions yet, but there's one point especially that I want to make, and it could be that all of these will be A, and these will be B, and these will be C, and this will be D. Is there another slide? Then I will, so that's, I'm done with that. I've decided what your grade is. Then I will read the papers. And each of you that wrote a paper will have the chance to improve your grade. And for example, I'm not so sure. Yeah, so for example, if you were one of the people that got a grade of 75, and you got 15 points on the paper, that would have, remember it's a 20-point paper, that would pull you up to 90 points, and that 90 points would in turn have pulled you into the A category. It will be nice if it always works that way, but it won't necessarily. Sometimes you will have, in this case, for example, a grade of 55 on the quiz, and you get 15 points on the paper. That would just pull you up to a 70, and that would leave you in the same grade category. So I hope those 2 examples give you a sense of what I'm going to do. The point to remember is that they are independent events. I'll do the curve independent of the papers, and your grade will then be determined by that curve. But then you'll have the chance to pull your grade up with the paper, and that may, in fact, I hope this is true for everybody, that may in fact pull you over a grade line, and you would end up there. Just in looking at the quizzes and looking at the papers, I glanced through the papers very quickly. You've done very well, and I very much appreciate the fact that you've done well. You did well on the quizzes, and you did well on the papers as well, and I appreciate that, and thank you for that. So now we go on to the lecture. Oh no, we're going to do the quiz now? Okay, we'll do the quiz. Okay. And then we'll go on to the lecture. Okay, why don't we get at this very interesting but short story on stem cells and why they have been controversial and why they're interesting and why people that do research on virtually any kind of human disease, although it would work well for non-humans as well, are interested in being able to use stem cells. Remember, I mentioned the fact that they were not that researchers in the U.S. were not able to use government money to do research on stem cells. So let's see what are stem cells to begin with. We start with a process that you're all, I expect, all familiar with from your high school biology, and that is that we're talking here about fertilization of the eggs. The eggs are released, and very shortly after eggs are released into the oviduct, sometimes called the fallopian tube in English, they are fertilized, and that is because sperm come up through the vagina, through the uterus, up the and fertilize the egg here, and then the egg continues its path down to the uterus where it implants. And as it's continuing its path, it's dividing. So the fertilized egg is already it's a potential human being because it's been fertilized, and it's a unique human being, a single sperm, single egg, and it's already been dividing. So these divisions here are happening during this trek down to, before it ever gets to the uterus where it implants. Now there's the unique thing about these cells that make up, this is the, I don't know if that's intended to be the 4-cell stage, and this is the 8-cell stage and the 16, but in any case, these are the beginning, the first cell divisions that occur, and there's a very unique thing about these cells that you don't find anywhere else. You find something that comes close, so people will talk about stem cells from other tissues, but these are very unique in that every single one of them has all of the genes available, and every single one of those genes can be turned on. Now you know that that all of the genes are in all of the cells. You know that. We've mentioned that several times, and you probably learned it before you ever came here. So all of the genes are in every cell. The difference is that a cell in your liver, let's say, can only, there's a small number of cells in your liver that can become new liver cells, but they can't become skin cells, and they can't become heart cells, and they can't become anything else. So most of the genes have been turned off as the cells develop. But at this stage, all the genes are, they're not all turned on, but they're all capable of being turned on. And they are called stem cells because they are the first cells in the development of any particular kind of cell. So that's the important thing to remember about these cells, and that's why researchers are so interested in them. For example, let us say the researcher that works with nervous tissue, and we know that there are serious automobile accidents, for example, that hurt badly nervous tissue in the backbone, and it can cause paralysis. Pardon me, it can cause paralysis, and it is something that we have been working on for a long time. We physicians and so on, people that are researchers. Now, the problem is that in the spine, there's no capability of those cells to reproduce themselves. So you get damage in the brain, it doesn't reproduce. It can't make new brain cells. So what the researcher says is, wow, here we have a bunch of stem cells that could be used to make these cells that can repair damage that we haven't been able to repair before. So these are very interesting to researchers. They would love to have lots of these cells in their work with any kind of reproduction of the cells that has to be done because of illness or because of disease or because of traumatic accident and so on. Now, I started out by saying, here they are, they're coming on down, they implant in the uterus, and then it grows into a baby. So where do we get these? Where do we get these is the question. And the answer is that while these stem cells, first of all, they can reproduce themselves, and second, they can become all kinds of different things as the embryo develops. They can become skin cells, they can become organ cells, they can become everything that the body needs, those stem cells can become. But as they develop, they increasingly shut down all of those other genes that are not related to this final stage here, whatever these stages are. And they aren't named, but this could be liver, and this could be heart, and this could be skin, and so on and so forth. And as they move toward the end of their development, all of the other genes get shut down. They're still there, the genes are still there, but they aren't functioning. They're genes that are turned off, and they're turned off permanently. It's a very curious thing about development in cells. So this this abbreviation here stands for human embryonic stem cells. As I mentioned before, there are some other kinds of stem cells, but the ones we're talking about are the embryonic stem cells, the ones that come from the embryo. And we still haven't answered the question about where they come from. Where do we get them? This we just talked about, that is to say, genes that a nerve cell needs to continue to be turned, they will be produced in the nervous tissue, but all of the other genes are shut off. Stem cell, I'm just repeating the same lesson here a couple of times. Stem cells can become blood cells of different types. They can become nervous tissue, nerve cells of different kinds. They can become liver cells. The question is, again, where do they come from? And this is where they come from. And we'll get into a little bit of detail here. There are increasingly couples who want to have a baby that cannot. That no matter how hard they try, they don't. It doesn't happen that that the egg is fertilized. The mother's egg is fertilized by the father. And so techniques have been developed. Well, first let's answer another question. Why is it that is increasing? It's increasing because women are waiting in a longer and longer period of time before they go about having children. The maximum fertility is actually at about age 16. Now, if you think about evolution, you can understand why it would be so young. But more and more women are waiting to have babies. Women and men are waiting to have babies. And so the average age of the first child is getting older and older. I don't know what it is right now, but I do know that it's not at all uncommon for women to have their first child when they're 30 or older than 30, sometimes up to age 40. So they're waiting. And they're waiting for a variety of reasons, but primarily they are establishing themselves in their careers. Women are twice as likely to have a career track now as they were 20 years ago. So that's changing. And so women are waiting. They're waiting longer and longer. Well, that's fine, and there are all kinds of good reasons for that. But the likelihood of one of their eggs being fertilized is going down as the years pass. So that's kind of the chance you're taking when you delay having children. So because that's happening, there's an increasing number of couples who just simply can't seem to, the mother can't seem to get pregnant, or the couple can't seem to get pregnant, I guess is the right way to say it today. So what do? Well, it's called in vitro fertilization, and it's called in vitro, all in vitro means is in glass. So here's how they do it. The female is given hormones, and that causes her, instead of just the one egg that she might produce in each cycle, she produces many eggs. Those eggs are harvested and they're put into a petri dish. Sperm is then gathered from the male, from the father, and that sperm is put in the petri dish as well. And things are done in such a way that the fertilization of the egg in the petri dish is more likely than the fertilization of the egg in the fallopian tube, in the oviduct. So that fertilization takes place, and then they take the fertilized egg, and it's actually more than one, and they implant it into the uterus. So they've got a fertilized egg, they implant it in the uterus, and very often it's successful. Sometimes a couple has to try several times to make this work, but it very often works. There are a couple of difficulties with it, though. One of them is that to make sure, or to help increase the chances of fertilization, they will take, the docs, the physicians, will take the fertilized eggs and implant them in the uterus, not just one, but maybe they'll do 4, with the hope that one of them will implant. Now, you know, it's kind of a guess, and sometimes many more implant in the uterus than was planned. There was, I don't know how long ago it is now, it must be a year ago, a mother who had octuplets, is that the right thing? She had 8 babies in the U.S., and in fact, she's referred to as Octomom because she had these 8 babies. And this was why. Her physician put a lot more than 1 in. Nobody knows how many he put in, but he certainly put in at least 8. And there was a high percentage of implantation and growth, and so she ended up having 8 babies. Having multiple births has become more common, and in fact, whenever you hear about somebody that had twins or triplets, one's first question always is, did you have in vitro fertilization? Because that raises the chances of having multiple births. So that's one problem, the problem of multiple births and the fact that you have to kind of guess at what's going on. Now, other people will abort. So if they only want 1 baby, they will have if they've got more that are implanted and growing, they will abort all but 1. Not very many moms do that. Not very many mothers do that. Not very many parents agree that that's a good thing to do. But in any case, so that's a hard decision. So that's one thing that has to be thought about. Here's another one, and now we're getting closer to why research was not supported with these stem cells. And that is that the extras, let's say that you've put everything into the petri dish, and you've got fertilization happening, and you end up with 15 eggs that have been fertilized. Well, you don't want to, even if you put more than the one you want to implant in the uterus, you do not, there are some you're not going to use. There are some you are not going to use. What do you do with them? Well, let's come back to the fact that a fertilized egg in a situation like this is a unique individual, a potential unique individual. It's a specific egg that's been fertilized by a specific sperm, and that fertilized egg is destined to become a unique individual. Not any old individual, but a unique individual that's the product of that sperm and that egg. So they're saved, and that's one of the reasons that they're saved, because they represent potential unique individuals. Now, one thing that the parents could be thinking about is, well, it's great, we've got one going here, but what if it doesn't take? What if the one that we've implanted, what if I miscarry? what if a miscarriage takes place? What do we do then? Well, what they have here are some extras that could be used, rather than go through the whole process again. There are some extras that could be used to repeat, to try again. So that's one thing. Another is that there are people who want to have a baby, but for one reason or another, neither or both of the parents, maybe they're both very old, maybe they're very old, maybe they're 50 years old, and they're past the age of having a baby that is uniquely belongs to the two of them. So they might, in fact, purchase these embryos. That's another possibility. So there are reasons to say this. The main reason, though, is that they are considered to be living organisms, unique living organisms, and so they are saved in liquid nitrogen. It's a good thing, depending on your perspective, that can happen when you have embryos at this stage, 8, 16, 32 cells. They can be saved in liquid nitrogen and then carefully thawed and used. They're not killed in liquid nitrogen. So there are hundreds of thousands of embryos that are in liquid nitrogen in storage places, all from this artificial fertilization, and they are all available for researchers to use. But these do not belong to the researchers. They belong to the parents. And so the parents are the ones that have control over them. And furthermore, it's been determined, or it's been decided that only the parents can decide whether or not these might be used for research. So they would have to say to themselves, okay, it is a unique individual, and if we implant it in the uterus, it could grow into a unique individual, but we are going to trade that opportunity for the opportunity of these cells being used in research. So what happens in the case of research? This is an actual embryo, and it happens to be at the 8-cell stage, so those are all stem cells. They all have this great capability, and one can go in there with a tiny microscopic forceps and pull out one of those cells, just like is happening right here. Pull out one of those cells and do the whatever kind of research you want to do. You may want to put that, you may have circumstances where you know you are highly likely to be able to get that cell to develop into nervous tissue. And if you can do that, then you have a great tool for dealing with nervous tissue problems. This is how far it's gone. People working, these are rats, they aren't human, but everything's more or less the same. And people have been able to take these stem cells and use them to develop, to grow lungs. And these lungs are the actual size of lungs that are in a rat. It wasn't just the stem cell itself, they had to give the stem cell some help. But they could not have done this without this help, without the addition of the stem cells. So people that are interested in growing tissue that they couldn't otherwise get are very interested in stem cells. And this came out of a popular magazine in the United States, Time magazine. But you can find where the work is reported by going down here if you want to pursue this. But it's a very good example of how stem cells can be so useful and so unique in the research that different people are doing. So, really a pro and a con kind of circumstance, right? There's an election going on in this country, but there also happens to be an election going on in the United States, and one of the candidates said he had a very important opinion about this, or important in that it made people know where he stood on the issue. He said, I believe that life begins at conception. Life begins when the sperm fertilizes the egg. That's what I believe. And therefore, if one would kill these cells that result shortly after fertilization, that is abortion. And I cannot tolerate abortion. I do not believe in abortion. Again, this is a candidate by the name of Kane, one of the candidates in the election. He's run into a little trouble, and I don't know if he'll last very much longer on other accounts, but I don't know if he'll last very long. But he made that very strong statement. And that was the statement that Bush was making when Bush said, I'm not going to let any federal funds be used for this kind of research because it's abortion. It's abortion, and I don't believe in abortion. So it's not going to happen on my watch. So I had these strong feelings. Now, what are the opposing feelings with regard to that abortion issue? Well, there are people that don't believe that this is really an individual that we're talking about. Those people will say, no brain. How can it be an individual without any brain? No nervous tissue, no brain. It's not an individual. It's not an individual until it has some awareness in some form or another of the world around it. So that's how some people think about the abortion issue. Different people have different ideas. You might be aware that there are lots of people, in fact, that think that abortion is perfectly okay up through the third month of pregnancy. So different people have different views about abortion. But those that feel that it's abortion, if you do anything to harm the embryo after the egg has been fertilized, those are the people that have trouble with stem cell research. And I think that one of the problems that we in the U.S. have, anyway, with discussing this is that we don't talk about that detail of it. We just don't talk about it. We don't talk about the fact that for some people, at least, it's an abortion issue. So that's stem cells. Simple with regard to the biology, very complex with regard to the morality of it and people's feelings about whether or not it's ethically sound and ethically okay. Now, the University of California has been pushed to take a stand on using these cells in research. I mean, are they going to allow their people to do research in this general area? And the answer is yes. And here's the statement that the University of California put out about 2 years ago, maybe closer to 1 year ago. The University of California believes that it is imperative that the scientific community be permitted to move forward with embryonic stem cell research that provides hope to millions of patients and their families. These are all of the people that are facing horrible, horrific diseases that stem cell researchers say they might be able to solve. So if you have a child, for example, that has one of these terrible diseases, and that child is headed toward death, unless some cure comes along, and a stem cell researcher says, I've got a potential cure, then you're going to have a different view about all of this. It's your child that's going to die. Is that okay? Versus the stem cell, the fertilized embryo that's going to die. So it gets to be an even more complicated issue, one that goes beyond just whether or not you believe in abortion. So that's where we are in the U.S., and that's where we are in the world. The European countries, most Asian countries, more or less align with what I've just described to you. It's accepted that this is a moral dilemma and that a decision has to be made, very often a personal decision has to be made in this process. It is just not as easy as it first seems, and it's not so easy as the simple biology of it would indicate. Simple biology, difficult problem, difficult ethical, moral problem. that's it. Thank you all very much. I appreciate the fact that you all kept coming. This could be a half-full classroom, and I'm glad that you're all here. Thank you all very much. I enjoyed it. I hope you did as well. Thanks.